Welcome to the Plant Physiology Photosynthesis Workshop. This is a workshop that is associated with the topic plant photosynthesis, which is part of the plant physiology subject that is taught within the Bachelor of Agriculture and Technology. This degree is offered both at North Melbourne Institute of Tech and Melbourne Polytechnic. Please visit our website www.nmit.edu.au for more information on both this course and other courses that we teach. My name is Dr. Nikki Cooley, and I have constructed this workshop with Dr. Alistair Reid in the aim that our students would understand photosynthesis and its associated mechanisms and structures. The aim of the Photosynthesis Workshop is to improve and enhance your understanding of photosynthesis. Photosynthesis is an important component of plant growth and development, and an important component of understanding this subject. So far, you have been given a number of lectures and recommended reading, along with 3 associated tutorials. In this workshop, we will start by Dr. Reid giving a 10-minute review on the good and the bad of photosynthesis. This is not included in this video. We will then follow by the results of a modelling exercise. And finally, we will present the exam question. This is a way of testing your understanding to date. In order for you to prepare for the photosynthesis workshop, the first thing you will need to do is watch the lectures, do the associated reading, and finally watch the photosynthesis video on the light reaction, Calvin cycle and electron transport. The images and information presented in this video will use the base of the modelling exercise. Modelling is a scientific technique that is used to test and develop the understanding of a concept or mechanism in biology. In the exercise today as a class, we will model the process of photosynthesis. The class will be separated into 6 groups, where each group will be assigned to a model, and the model will have specific structure or stages that require model subcomponents. Each model subcomponent will have a specific specifications. These are listed below. The aims of the exercise are for you to demonstrate your knowledge of photosynthesis, demonstrate the important relation between structure and function in this mechanism, describe how electrons become excited, how adenosine triphosphate and NADPH are generated, how energy compounds are used to produce G3P in the Calvin cycle. The first exercise was assigned to Group 1. They were to model the chloroplast. The model specification for this was to model the structure of the stacked thylakoids in the chloroplast. Their model should ideally illustrate the large surface area of the thylakoid membranes. They should demonstrate the thylakoid and the stroma structure and the transfer of energy. We suggested using water as the stroma structure. Please watch their video now for their interpretation and presentation of their model.
This model illustrates a chloroplast. It contains thylakoid membranes. The parts of thylakoid membranes are lumen, which is this green stuff you see here, and there is the stroma, where the space is, and outside here is the outer membrane, and in there is the inner membrane, and the yellow thing you see there represents the NADPH, and the red color represents ATP. Yeah, that's it.
Group 2 were to model Photosystem 2. The model for specification for Photosystem 2 was to represent the chlorophylls and the chlorophyll reaction centre, that the Photosystem 2 sat within the thylakoid membrane, the process of electrons, the transfer of ground state electrons to an excited state. This model was also to illustrate how water was split into its various components.
Okay, so we should start from Photosystem 2 model. Light from the sun, the rays are at 680, and protons in the rays land on grounded electrons. Once it lands on the grounded electrons, the electrons become excited. The excited electrons move into the chlorophyll reaction center, which is here. In the center, H2O is split into oxygen and hydrogen. The excited electrons then, two at a time, move out of the
Model 3's aim was to model the electron transport chain. The model specifications were to demonstrate that the electron transport chain sites are located in the thylakoid membrane. Demonstrate how electron transfer occurs from the Photosystem 2 chlorophyll reaction center.
So this is a model of the electron transport chain.
It's located in the thylakoid membranes.
The purpose of Model 4 was to model Photosystem 1. Model specifications were to show how the electrons became excited and that Photosystem 1 sits within the thylakoid membrane. Show how NADP is synthesized into NADPH. Illustrate low electron excitation and high electron excitation.
This is a model of the electron transport chain.
It's located in the thylakoid membranes.
Okay, so we'll start with Photosystem 2, which is just over here. Now in Photosystem 2, light hits the Photosystem 2 system. At that point, photolysis happens, which is basically shown down the bottom here. So water is split to give oxygen and an excited electron there. The electron then is carried up through into the cytochrome via the protein here shown as the brown. So basically up through the green, comes up through the brown protein and into the cytochrome next door.
Cool. So cytochrome oxidizes plastohydrogenone and delivers electrons to Photosystem 1, which is the secondary one.
He want to reduce NADPH plus to NADPH in the stroma through femiluxins and FNR.
And finally, the last one. We are doing the transportation from the NADP to NADPH. So we got the electron from photosynthesis 2, which is from the low energy, which will be transferred to the photosynthesis 1, and then the energy will be absorbed to the protein, and the NADP will absorb and then change the energy to NADPH. That's it. Thank you.
The objective of Model 5 was to model the ATPase pump. The model specifications were to illustrate that hydrogen ions will go into the pump and hydrogen ions will come out of the pump, and this will enable conversion of adenosine diphosphate to adenosine triphosphate. The pump will be illustrated, the location of the pump, will be illustrated in the thylakoid membrane.
So this is a model of
thylakoid membrane. So basically what we've got here is hydrogen ions down here, represented with the green. So basically those hydrogen ions are pumped into the lumen when the electron transport chain pumps them through. So basically these hydrogen ions here, because they've got a lower concentration gradient, they travel up through the protein, and as they're doing that, they power the conversion of ADP into ATP. So that ATP is then taken to the Calvin-Benson cycle and is used to reduce carbon.
Thank you.
Model 6 was the final model in our modelling 3D exercise. The objective of Model 6 was to demonstrate the Calvin cycle. The model specification was to show that the Calvin cycle requires energy. In this model, it was essential that the correct number of carbon and energy molecules for each major step in the cycle was shown. It was important to show the number of carbon molecules and how these change as the cycle goes around. It was also important to demonstrate that this was in fact a cycle and that there was a regeneration part of this cycle.
Oh, so one person can just do it.
We're doing the carbon cycle, which is a series of reactions that reduce CO2 to produce the carbohydrate glyceraldehyde 3-phosphate.
Step 1 in the cycle is carbon fixation. CO2 is attached to ribulose 1,5-bisphosphate and then splits into 2,3 carbon molecules.
Step 2 is the sequence of reactions using electrons from NADPH and ATP to reduce CO2, which then form ADP and NADP+.
Step 3, the final step. Glucose 1,5-bisphosphate is regenerated every 3 turns of the cycle, 5 molecules of glyceraldehyde 3-phosphate are used to... completely lost there.
No, you're doing good. Carry on.
Of glyceraldehyde 3-phosphate are used to reform 3 molecules of ribulose 1,5-bisphosphate. The remaining glyceraldehyde 3-phosphate goes into the production of glucose.
There are 3 photosynthetic tutorials that I would like you to ensure that you complete. The first is tutorial 7 on the light reaction. The second is a data interpretation exercise on the effects of pruning, rootstock variety and water stress on vine carbohydrate partition status. This is tutorial 8. And the final tutorial to complete on the topic of photosynthesis is tutorial 9 on photosynthesis D-carbon reactions. Tutorials 7 and 9 have been covered in class. Tutorial 8 has not. Attempt the exercise and bring along to your next tutorial. That brings us to the end of the 3D modelling exercise. For those of you that were unable to attend the workshop, it might be a worthwhile exercise for you to attempt in one of the stages. Compare your attempt with the video shown or constructed by the students. The second part of the workshop was to test your understanding and knowledge of the photosynthesis, both the light requiring and non-light requiring reactions. I have compiled a short answer open book exam question for you to do this. The next stage of the workshop is to prepare for an exam question. In the preparation here, you will be required to watch 2 videos. The first is on biomimicry. These are where you learn lessons from nature and apply them to a man-made or constructed concept or idea. After this, please watch the video on nanotechnology. In this question, ideally give yourself 15 to 20 minutes to answer. Write your answer as dot points. We will be reviewing the answers for those that were not able to attend the workshop on Monday the 12th of May tutorial. question to attempt is, the efficient and sustainable production of energy is a significant 21st century problem for mankind. A potential solution to this problem would be to apply biomimicry. That is, our understanding of photosynthesis to energy production. It is your task to list areas of the process of photosynthesis that might be applied to the design of an artificial photosynthetic cell. That brings us to the end of this workshop.